4 to the current exam before they go on D2L. So by the end of the weekend, your exam 2 score on D2L will be updated with that adjusted score. But then remember, there's still a point coming back, and the points for quizzes come back at the end of the semester. So that'll be a tally, but then the 4 points for this exam will be added for this one. So please know that. And yeah, if you do have questions or concerns, feel free to reach out. Happy to discuss whatever, go over your exam. But that's the situation with exam 2, and that's why things haven't been posted to D2L yet. So yeah, I think that's it for announcements. So this is our last section of the class. Gonna get into stimulants now. Yes, please.
Under there's like a section in D12 under quizzes. It says second multiple choice exam, and it like you can take it'll let you take it. I just want to make sure we don't have to do this. It's nothing to worry about.
Sure. Yeah, there's an announcement on D2L for that. Thank you for asking. That is another announcement I wanted to say again. So all exams for this class are in person. There has been something posted to D2L, but it's for a specific student who's notified. It has nothing to do with the class in general. Please know all exams for this class are in person. Anything you see on D2L is not outside of the quizzes, okay? Exams for this class are in person. Nothing on D2L is for anyone except for those who've been contacted directly. So please, if anyone asks, please tell them that. I'll put another announcement on D2L. Thank you for asking. All exams for this class are in person. Anything on D2L is a specific situation for a specific student. So any exam that you see under quizzes on D2L is not for the class in general. All exams for this class are in. Okay, we got it. You got it? Thank you for asking, Joe. Do want to clarify. Questions, concerns outside of that? Let's dive in. All right, amphetamine, related drugs. We're going up this time, right? This section, so brief history here. This is kind of an interesting one. Original stimulants came from natural sources, not a big thing to say. You know, using plants as medicine and that has been done for thousands of years. But this one is a little bit different. So, right, we talked about cannabis, we talked about opiates. These are molecules, well, THC, not just cannabis, right? They have a natural origin, but they have a target-specific receptor, right? And then they were isolated from a natural compound, and that's when they really got dangerous. This one's kind of interesting because there's stimulants in lots of different plants. The one used as an example here is ephedra. Ephedrine is the active ingredient there from this plant. But these, a lot of these were used in dual purpose or multi-purpose kind of thing. They weren't just used to kind of bring you up. They're decongestants, they're cough suppressants. They're used as those sorts of things in more traditional medicine. But outside of this, it's kind of interesting because it's sort of like the history is there was a bunch of mild things, and then there was amphetamine, and then everything changed, okay? So there was stimulants out there. Again, they're more dual or multi-purpose kinds of things, but the real stimulant drug, amphetamine, was the game changer. That after that was synthesized, it says in 1887, everything changed, okay? And it really has to do with, you know, medicinal chemists were trying to work on this, having isolated ephedrine, and trying to improve on its potency, if you will, and then cut down on the various affects that it has and really kind of hone in on specific applications for it is really what brought this about. But once amphetamine hit the market, I mean, everything changed. This really brought about a change in this space, if you will. And so as these kind of say, a lot of the interest in this were in militaries, understandably so, especially pilots. That was a huge group or occupation, if you will, that immediately was, took advantage of these. For understandable reasons. People that are flying a 20-hour flight, you can't really, coffee and tea really aren't going to cut it in your 15th hour. So something that's going to keep you awake and focused for something that's as involved as flying, especially those, earlier planes that really required a lot of attention to detail. But so there was sort of a lot of whatnot, and then there was amphetamine, and then everything changed, okay? It wasn't just for initial applications weren't just for the pick-me-up side of it. This is also used for people that had narcolepsy or problems with sleep, as well as for people who wanted to lose weight for obvious reasons that we'll get into. That's one of the things that, one of the early aspects, besides making people feel good and giving them lots of energy, they just didn't need to eat, similarly to that they did prior to starting to use this. But a big problem started to happen with the development of methamphetamine once the chemists just tacked that one methyl group on there, as we'll look at. It changed everything in this space. And then the problem really started when a lot of underground production of the drug, if you will. 20 years ago, you used to be able to go into a drugstore and buy all of the Sudafed on the shelf because pseudoephedrine is most of the way to methamphetamine. But you can't do that anymore. Now you got to go show an ID and you get one damn box and that's it. It's got to last you, whatever. That's okay, and that's okay. So, but that's kind of a brief, very chaotic history for this. What these do gives you energy, psychostimulation, euphoria, increased attention, right? Appetite suppression is kind of a nice side effect from this. More fun weekends is some of the space. There's a lot of debate with that one, if it's really working or not. But performance increases are another thing that amphetamines are used for. And then, as I said, there is actual medical symptoms that can be changed with these. Decongestion as well as cough suppression with some of these. All right, and then we'll talk about the changes in cardiac performance are part of the endurance that helps, but it's also part of the toxicities we'll see at the end of this. So it's all about feeling better. It's all about giving yourself more energy and better focus. But there's definitely limits to this. So, and it's all about dopamine, really, with these. So, as I brought up, ephedra is used in traditional Chinese medicine. This was a stimulant plant used and for decongestion and cough suppression for thousands of years. But then when more modern chemistry methods developed and found that this ephedrine was the active compound, and you could see then pseudoephedrine is just an isomer of this. This was then developed and can be used in the same space. But as you see, it's very similar to dopamine, right? It just doesn't have this catechol coming off here. But then compare that to amphetamine and methamphetamine. And as always, don't need to know structures, just showing it because it's interesting and a lot of people do think in these terms. But as you can see, the difference between amphetamine and methamphetamine is really just this methyl group off the amine here. And it's amazing what a different effect this has, even though they do occupy the same space in terms of bringing people up in that. Methamphetamine has a lot more durability and potential for abuse. Not just from this base compound of this active ingredient, it sounds like also adulterants from underground synthesis procedures are also tainting or changing the overall effect of methamphetamine compared to an amphetamine that's actually produced in pharma that is quality controlled and whatnot. So, but these are the main players in this space. How it's broken down, as you can see here, There's a number of different pathways, but just remember CYP2D6. That's the only one we really need for this class. Cytochrome P450-2D6 is the enzyme that'll break down stimulant drugs, amphetamine specifically. Don't worry about COMT. Again, just remember 2D6 in terms of amphetamine, methamphetamine metabolism. Now, the really interesting or the unique aspect of this drug or these drugs is how they're really taking advantage of a well-established system, right? So what we've talked about so far, especially cannabis, opioids, you know, THC targets CB, right, cannabinoid receptors. Opioids target opioid receptors. These are drugs that have specific receptors that then cause their effect. Here, we're just they're taking advantage of a system that's already in place, specifically dopamine signaling, but it's really all catechols, right? All monoamines are affected by stimulant drugs, and we'll talk about it in a bit. So, yeah, kind of a ground up build on this one. Here's our presynaptic nerve terminal, postsynaptic nerve, we got a receptor, we've got neurotransmitter and synaptic vesicles. These get released out of the synapse, bind to receptor, causes an effect, That's great. But what we has also talked about in this very next figure is how, you know, we package and we release, but then there's this reuptake, right? So we have this cycle of neurotransmitter that's important for, this is the life cycle of our neurotransmitter. Monoaminergic neurotransmitter in a vesicle, it's released out into the synapse, does its thing. Whatever's left over then gets pumped back up into the presynaptic nerve terminal by this transporter, okay? Here's our cycle. Now with these drugs, a couple of different things usually happen. Primarily, inhibition of the transporter is the biggest one, right? So these drugs will physically block our transporter and not allow for reuptake of the neurotransmitter. So this allows for prolonged signaling and greater signaling. You're going to get a larger concentration of neurotransmitter in the synapse for a longer amount of time. So both of these are going to affect the signaling greatly. Another approach here, or another way these drugs have an effect on this system is through binding to the transporter and reversing the flow of it, right? So rather than blocking it out in the synapse, they actually encourage this transport to become an efflux, so that you're not just releasing neurotransmitter out into the synapse with your vesicles, the transporter is going to be pumping things out too. So again, the overall effect here is a greater amount of neurotransmitter in the synapse for a longer amount of time. Overall effect is a greater amount of signaling in this space, all right? And then here's our precursor for these neurotransmitters. Tyrosine is the base molecule for dopamine, norepinephrine, and epinephrine. Serotonin comes from tryptophan, but that's also a monoaminergic neurotransmitter. So, but this is the big distinction here, right? We're not just targeting a new receptor with a new drug, we're actually taking advantage of something that's already in place and having an effect due to that. Oh, so, now this is, so this is the main aspect of how amphetamine works. So let's look at this cartoon of presynaptic nerve terminal, postsynaptic nerve terminal. We've got dopamine, our synaptic vesicles, gets released out into the synapse, can bind dopamine receptors. Residual dopamine is taken back up into the presynaptic terminal by the dopamine transporter, or DAT, and then it's either packaged back into a vesicle by VMAT or broken down by mouth, right? We've done all of that. But now, amphetamine's mechanism of action. So amphetamine can also get into the synapse, and it is a substrate for DAT, right? This is transported by the dopamine transporter up into the presynaptic nerve terminal. It's also a substrate for VMAT, so it can be pumped into a synaptic vesicle and therefore displace dopamine from that area, right? You just have a finite space in there. You can only put so many molecules in there. You start throwing dopamine in there, you're going to have to exclude some, you start throwing amphetamine in there, you're going to exclude dopamine that's in there. And then the last thing it does is actually inhibits monoamine oxidase. So MAO is inhibited by amphetamine. So by displacing the amount of dopamine here, it will, being able to be taken up into the presynaptic nerve terminal, displacing dopamine here, and then preventing its breakdown, the overall effect is a larger amount of dopamine out in the synapse. Okay, so once again, higher concentration of dopamine for a longer amount of time equals larger amount of signaling there. These are the three aspects of amphetamine mechanism of action that are important for us, all right? And amphetamine-like molecule, methylphenidate, or Ritalin as it's better known, is similar but very distinct. So same thing we just covered here, with the exception of methylphenidate just inhibits the dopamine transporter. It doesn't do all the other things that amphetamine does, but it has a very similar overall effect of amphetamine. Just by blocking the dopamine transporter, it will increase the concentration of dopamine and the length of signaling, okay? So a couple distinct mechanisms of action that have the same overall effect on the system, degrees of which you can toggle through dosing, but yeah, amphetamine is going to have a much more pronounced and distinct euphoria over methylphenidate, just due to the fact that it does more in the system there. But as this says, the methylphenidate will inhibit dopamine as well as norepinephrine and serotonin transporters. Yeah, we'll talk about that in just a sec, but that's the big distinction. Amphetamine can be taken up by DAT, methylphenidate will block DAT, okay? But they're both causing that overall increase in the dopaminergic tone in this synapse. Now, yeah, I use this one with caution. Don't worry about all the details for this specifically. It kind of contradicts what I said. The figure is more, yeah, I just really like the figure. This is from a study that shows some of these things. Just remember that amphetamine hits these three transporters. And the reason I bring this up is the relative binding affinities for these. So they each have a higher potency, or they have tighter binding, better binding at specific transporters compared to each other. Amphetamine has its high, so here's a key here, I guess. I hope this is straightforward. The drug will be, the best binder is the leftmost, right? We're talking greater than symbols here. So the leftmost, or the transporter that's closest to the drug listed here, is the best, where it has the highest potency, where it binds best. Then the one at the rightmost is the worst binding. So amphetamine and cocaine both bind dopamine. They have their highest binding there, right? And then there's various degrees. So amphetamine has really poor binding at serotonin transporter. Fluoxetine, right, Prozac, this is an SSRI, so this is going to have this sub-nanomolar, this is amazing binder, but it binds to serotonin way better than either of the other monoamine transporters, right? So just remember that. The SSRIs bind SERT, that makes sense. Fluoxetine included there. So, but the idea, though, is that they can bind to the other transporters. They can have a modular effect on these other transporters, but it's really going to be one that they're targeting. And when we're talking about stimulant drugs, we're really talking about dopamine. We'll talk about serotonin in just a bit, how there's an overlap there, but for the most part, these drugs target dopamine signal, okay? And again, just to reinforce that, I guess, the size of the arrow shows where they have the highest potency. So MDMA or ecstasy is going to be for serotonin. We'll talk about that in just a minute. So you can see, similar to amphetamine, methamphetamine being like dopamine, MDMA is more like serotonin, right? But then again, amphetamine, methamphetamine, highest potency at dopamine, slightly less at norepinephrine, but that's, yeah, it's a much lesser desired target, if you will. We'll have some similar effects, but yeah. And then mescaline, yeah, we'll get back to that. We do hallucinogens. Might put that on there, but. So, remember, yeah, these drugs have distinct potencies or distinct binding affinities at specific transporters, and the ones where they have their highest affinity transporter will be the system that they have their greatest effect here. So on MDMA, these are the distinctions here that are really important. Pay attention to this figure really well. So the MDMA is going to be our red boxes with a blue top to it, if you will. Receptors are yellow jobbers. Then we got VMAT, serotonin transporter, and then serotonin itself is just a blue square. So here too, MDMA, similar to amphetamine, is a substrate for the transporter in particular, right? So MDMA can be brought up in the presynaptic nerve terminal. Here it will inhibit VMAT storage of serotonin, all right? But the biggest thing, so yeah, this will cause an accumulation of serotonin, similar to what we just saw with amphetamine. But the biggest distinction here is MDMA actually causes the influx of SERT to change it to an efflux, right? So it changes the directionality of the serotonin transporter to pump serotonin out into the synapse. So you're not just releasing serotonin by synaptic vesicle, you're actually pumping it out. So it's kind of an interesting thing. You know, one approach, you're blocking transporter and allowing lots of stuff to stay out, and then the other approach is you're just flushing everything out of the terminal, right? So you're not necessarily blocking the reuptake, but you're overcoming that because you're pumping so much more out into the system with this transporter reversal here, all right? And then very similar situation in terms of the effects here. The euphoria and reduced appetite are two immediately known or recognizable aspects of MDMA usage. The increase in confidence and social ability is somewhat unique. People do experience that with amphetamine, but there's when people describe the effect of MDMA compared to amphetamine, there's always a mention of warmness that you will get with MDMA that you don't get with amphetamine. It's not just a heightened sort of euphoria and emotional state. There's a warmness that you feel interacting with those around you and things around you that's unique to MDMA. Understandably, they call it ecstasy, but that's, yeah, one of those, I don't know, anecdotal things. Some of the bad aspects of it we'll get to at the end of this. This is kind of an interesting one because it really changed after this became, the drug got in vogue, if you will. So dehydration was one of the initial problems that people had with all stimulants, especially MDMA. And there was something about you were so busy having a good time, you just didn't want to drink or didn't really think about it, especially in terms of people using it as a club drug, if you will. You go to a club, it's really hot, everybody's dancing, the music's all loud, and you just go and go and go, and you don't think about it, and the next thing you know, you're dehydrated, and then you start to suffer the effects of that. That was one of the main problems. Again, we'll get back to this. But interestingly enough, this started to be known over and over again, or there were multiple cases of people using this drug, getting so clinically dehydrated they had to be hospitalized for it. So the word started to get out that if you're using MDMA, remember to drink. And so people really took to that and they wanted to be healthy. And it was interesting because then what happened was people started doing the exact same things on ecstasy as they were doing initially, but they were just drinking a lot of water. So then the problem became, when you sweat, you're not just dumping water out, right? That's waste products as well as a lot of salts. A lot of electrolytes come out then, and that's the problem that they started happening then because people were drinking a ton of water but not replacing electrolytes too. So they would become hyponatremic, where you have too low sodium levels in your body, and then that causes a whole kinds of problems because people were then drinking excessive amounts of water but not balancing it with electrolytes. So it's kind of a compounding problem, if you will. It was good that people that were using this chronically were listening to the medical community and trying to respond to it, but a lack of really understanding of what they're doing. Right, so with that hyponatremia comes a lot of muscle aches and pains, and then it can get very severe if you get overly hydrated but have depleted electrolyte levels. So, anyways. Okay, we keep showing this in different ways in this class. Dopamine pathway, right? Here's our mesolimbic, mesocortical portions of our dopamine pathway originating in the VTA, projecting into and through the nucleus accumbens and then out into the PFC, right? And keep talking about this increased dopamine presence is what will give rise to euphoria, but the reason that I'm bringing it up now is because it's really the increased dopaminergic tone in the nucleus accumbens really drives this euphoria. And I'm going to say this now, and we can just kind of, I really don't like to make it a big part for this class because it's such a nuance in the field, but it should be said because, the more that we understand about dopamine, it really isn't the feel-good neurotransmitter. It's been labeled for generations as that, and it really isn't. It really is a neurotransmitter that says, there's something really interesting here, I need to pay attention to it. All of the euphoria and good feelings and stuff come from an elevated dopamine level, but it's more the, like the endogenous opioid, beta endorphin and those sorts of things that really do cause good feelings in our blood that are happening. It's also, if you are, if anybody here has ever set their mind to doing a project and you had to focus on it, you had to do a lot, you may even have to sacrifice your time or something to get it done, but then you get it done, there's a natural reward system in our brain that gives us rewards for doing that sort of thing. And that's more of the reward you can get from these drugs with the idea that if you're able to be more productive on them, that you'll get more rewards because you get more done, okay? That's enough soapbox stuff. What I'm just saying here, it is all about dopamine. It's about elevated dopamine in the nucleus accumbens, but it's always good to know that nuance in the field. It really isn't the dopamine that's causing the euphoria. The dopamine is saying there's something awesome here, I need to pay attention to it. It's other cascades that are happening that cause us to feel good. But again, coming back to the class, elevated dopamine in the nucleus accumbens is what's going to set the stage for the good feelings of euphoria that people experience on these drugs, all right? How does that sound? Is that how are we? I don't mean to be contradictory or intentionally vague. Regardless of what my wife says, I don't mean to be intentionally vague. I really do want to not. Okay, so along with that, or how we do know that, by molecular studies of where things bind in the brain. So looking here, amphetamine treatment was associated with a decrease in raclopride binding in the nucleus accumbens. Raclopride is an agent that can be used for detecting binding of dopamine, so it'll bind to dopamine receptors, and it can be displaced by endogenous dopamine, all right? So it's a nice little tracer because you can see where dopamine receptors are in the brain, but then you can use an endogenous agent like dopamine to displace it for studies. It doesn't bind so tightly that you can't do that. And as these fMRI scans are showing, these areas, the nucleus accumbens areas, are the hottest with this raclopride. This is a radioactively labeled raclopride. So, as it says, raclopride binding is correlated with drug wanting. So in this study, they had people respond to cues and describe the amount of drug wanting they were experiencing at any particular time. And as you can see, these two plots that were, well, the one on the left is the most important here. So the amount of raclopride binding in the nucleus accumbens. On this subjective scale, negative values were associated with less drug wanting, positive values are more drug wanting. The more drug wanting you have, the more raclopride you have binding in the nucleus accumbens, okay? So it's just reinforcing this idea that it really is the dopaminergic tone in the nucleus accumbens that's driving the effect of these stimulant drugs. So, yeah, the last note, the drug wanting is correlated with exploratory excitability and novelty seeking. These are personality traits associated with thrill seeking, if you will. And those sorts of personalities tend to be more prone towards development of addiction based off of their really natural brain wiring. But that's, yeah, kind of one of the endpoints there. However, I really wanted to communicate here, it's all about dopamine. It's all about dopamine in the nucleus accumbens. You can show that through raclopride binding. And yeah, that's. So, here's one interesting aspect of stimulant use that is important for us. CART peptides, cocaine and amphetamine related peptides, all right? These are really interesting because we didn't know them. Well, as the name implies, we didn't know about them until people started using these stimulant drugs. I mean, to jump ahead a bit, we found out these are naturally expressed peptides in the brain for certain, they're clearly some kind of survival instinct or just some communication from the brain to the rest of the body. But we didn't know of them until we started researching people that were using these drugs, hence the name cocaine and amphetamine related transcript. Okay, but as it says, these are synthesized in areas of the brain targeted by amphetamine, and one of the aspects of CART that's important is this can inhibit feeding behavior caused by NPY. All right, so hopefully everybody's okay with this slide coming back around again. So we're talking about stress relief and we're talking about alcohol. Pro-stress peptide is our CRF, and that binds to its target receptor, on and on and on and on and on, and then it upregulates NPY, right? So NPY is the anti-stress peptide, and NPY production is affected by chronic alcoholism, right? But what can also interfere in this is CART peptide production, all right? So part of why this does what it does goes to our autonomic nervous system, right? We've got sympathetic, parasympathetic. Your sympathetic is the fight or flight response to everything, keeps you alive, lets you know that there's a threat around, makes you respond to it. On the other side of things, when everything is cool, your parasympathetic is the feed and breed response, right? That's when you can cool out, get something to eat, have some fun with somebody, or more than one buddies. But just, those are the two essential sides to our autonomic nervous system. NPY is going to induce feeding as part of its response, right? And part of its de-stressing a body, it encourages feeding, where CART peptide will interfere with that, as we'll see. But as it says, CART peptide regulate reward, feeding, and stress response, okay? So if we actually look at an in vivo experiment, an observation chamber here, we've got a mouse inside, and we're doing our bar press for food, right? You press a bar, you get a pellet of food. Throw an animal in a chamber, and it's going to do that a certain amount of time. It's going to just explore the chamber, and then once it correlates a bar press with a food, it's going to do that a certain amount of time, right? So if we plot bar presses as a function of treatment, our control treatment is going to have a certain amount of bar presses. If we dump NPY onto this poor animal, it's going to increase their desire to eat, right? So the overall effect is that you have an increased number of bar presses because they're going to want to eat more than just the control groups, right? Now let's throw CART into the mix, and it's going to abolish that. CART is going to return this back to the basal level control, meaning that CART will abolish NPY's effect in wanting to eat, okay? So you got an animal, it's going to want to eat. You throw NPY on it, it's going to want to eat more. You throw CART in there, it's going to ablate that effect of the NPY. And they know that this is a CART determined phenomena because if you put the exact same situation we were just looking at, but you also dump in an antibody against CART, so a CART inactivating antibody, this effect is wiped out, as shown in this further hypothetical data here. So when we had CART plus NPY, we just had the normal amount of feeding. CART plus the antibody inactivating CART plus NPY is going to go back to just the NPY level of feeding, meaning CART will interrupt the effect of NPY, but CART's effect can also be interrupted and allow NPY to have its effect, all right? How does that sound, everybody? Straightforward, hopefully. Cool. So, the bad side of things, though, we talked just a little bit, this does make you feel good and it can have a lot of effects. But one of the biggest toxicities from amphetamine use comes from its activity in the norepinephrine space, all right? So, norepinephrine is an endogenous ligand for adrenergic receptors, both alpha and beta style. So this is looking at exactly what we just looked at, presynaptic nerve terminal, postsynaptic nerve terminal, here's receptors, and this time we're talking norepinephrine, all right? Norepinephrine will bind to beta style receptors and cause an increase in heart rate there, all right? The same time, at alpha receptors, it's going to cause an increase in the vasoconstriction of blood vessels, right? So these are usually in different areas. These are usually happening at different times. The body's really good at tightly regulating these kinds of things because if you think about both of these phenomena, you don't want them happening at the same time. Because in one situation, you're increasing the heart rate. In another situation, you're decreasing the diameter of the blood vessels. So you're going to be pumping more blood through thinner vessels. That increase in pressure is going to come back and it's either going to blow out your pump or it's going to blow out the pipes. So this is a big problem here where people have these adverse cardiac events. And this is prolonged usage, you know. This is specific situations. This isn't you get a prescription for amphetamine from your doctor to help you focus and next month you're having valve work done. This is a long time coming, you know, many decades with abundant use of these, but this is where it comes from. It really happens when you have an overload of stimulation at alpha and beta adrenergic receptors, causing an increase in blood pressure through heart rate, and then a constriction or vasoconstriction, causing the vasculature to shrink. So, yeah, that shift in pressure causes all kinds of pathologies and bad. So, that's amphetamine. Another aspect of methamphetamine is a bigger one here that you can have is actual breakdown of biological tissue, especially in the brain. So dopamine here, as shown here, in certain instances, this can change from one form to a highly reactive form in its quinone form, as shown here. Okay, these quinones are highly reactive compounds that can interact with all kinds of organic material. And when it's in the brain, it becomes very sensitive. So not to spend a lot of time with this, the idea here is there's several different scans that show brain activity. And we've got control and then chronic meth user. And what you can see in all three of these, so red and orange are associated with higher levels of activity in these brain regions. And then green and blue are lower activity. As you can see in each of these three instances, in the methamphetamine, chronic methamphetamine treated brain, there's a dramatically decreased amount of brain activity that's correlated to destruction of neurons in those areas. And then it was quantitated here, quantified over here, so tyrosine hydroxylase activity, and then just overall dopamine. Comparing saline controls to methamphetamine, you can see a drastic reduction in the amount of matter there and functioning, all due to chronic methamphetamine overuse, where dopamine is converted into its quinone form, becoming highly reactive and just destroying tissue. I mean, without regard to what it is, really. It's kind of like bleach at that point. It just starts reacting. So, bad. And lastly, MDMA. So I discussed this already. Initially, hypothermia was a big problem just due to the fact that people were getting dehydrated, and a lot of that came from overproduction of free radicals in their mitochondria. You know, you need those terminal electron acceptors, if you remember from cell biology or wherever you had metabolism study. Those ATP pumps need to have somewhere to dump those electrons on when they're spinning around. That's what you need water for. If you don't, then you can just kick out a lot of free radicals that will then do damage to everything you come in contact with. But that was a big aspect with MDMA toxicity, was that overabundance of free radicals generated by overactive mitochondria without any acceptors coming in from water. So that's bad. And I think that's, it's ended on a high note. So this is a study, Global Burden of Disease, from a handful of years ago, looking at amphetamine use disorder death rate. And the good old US of A is #1 in the whole world. So, way to go. Anyways, have a good weekend, everybody. See you Tuesday. Let me know if you have questions. What's happening?